Microbiology and Parasitology for 5th semester students of the Faculty of Medicine, Microbiology and Parasitology. Here is the detailed and structured explanation of microbiology and parasitology. Typically studied in the 5th semester of medical school. 1. Introduction to Microbiology and Parasitology. Definition of microbiology. The study of microorganisms, bacteria, viruses, fungi, and protozoa that can either cause diseases, pathogens, or benefit human life. Definition of parasitology. A branch of science that studies parasites, their hosts, and the interactions between them, including the effects of parasites on human health. Learning objectives. Understand the characteristics of medically relevant microorganisms and parasites. Analyze pathogenic mechanisms and host-parasite relationships. Learn about the diagnosis, prevention, and treatment of microbial and parasitic infections. 2. Microbiology. 2.1 Classification of microorganisms, bacteria, prokaryotic organisms categorized based on shape, cocci, round, bacilli, rod-shaped, spirilla, spiral. Gram-positive or gram-negative, based on gram staining. Viruses, infectious particles that require a host cell to replicate. Classified based on DNA or RNA, enveloped or non-enveloped structure. Fungi, eukaryotic organisms that can be unicellular, yeast, e.g., Candida albicans. Multicellular, molds, e.g., Aspergillus. Protozoa and algae, eukaryotic organisms found in water or soil, some acting as opportunistic pathogens. 2.2 Structure of microorganisms, components, cell membrane, cell wall, capsule, pili, flagella, and spores. Genetic elements like plasmids in bacteria that play roles in antibiotic resistance. 2.3 Mechanisms of microbial pathogenicity, adhesion to host cells. Toxin production, exotoxins and endotoxins. Evasion of the immune system, such as through antigenic variation. 2.4 Microbial infections, bacterial infections, examples, tuberculosis, Mycobacterium tuberculosis. Skin infections by Staphylococcus aureus. Viral infections, examples, hepatitis B, HBV, influenza, HIV, AIDS. Fungal infections, examples, candidiasis, Candida albicans, aspergillosis. Protozoal infections, examples, malaria, Plasmodium spp. Amoebiasis, Entamoeba histolytica. 2.5 Microbiological diagnosis, microscopic examination, Gram staining, Zeil-Nielsen staining. Microbial culture on selective or differential media. Serological and molecular tests PCR, ELISA. 2.6 Microbial therapy, antibiotics, for bacterial infections, such as amoxicillin for respiratory infections. Antivirals, for viral infections, such as oseltamivir for influenza. Antifungals, for fungal infections, such as fluconazole for candidiasis. Prevention, immunization, hygiene, and the use of antiseptics. 3. Parasitology. 3.1 Classification of parasites, protozoa, unicellular organisms, e.g., Plasmodium, causes malaria. Giardia lamblia, causes giardiasis. Helminths, worms, nematodes, roundworms, Ascaris lumbricoides. Trematodes, flatworms, Schistosoma spp. Cestodes, tapeworms, Taenia solium. Ectoparasites, parasites that live on the surface of the host, such as lice and mites. 3.2 Life cycle of parasites, direct cycle, does not require an intermediate host, e.g., Enterobius vermicularis. Indirect cycle, requires an intermediate host, e.g., Plasmodium spp. Needs the Anopheles mosquito as a vector. 3.3 Mechanisms of parasite pathogenicity, tissue damage, e.g., roundworms causing intestinal obstruction. Nutritional depletion, such as anemia caused by Ancylostoma duodenale. Inducing harmful immune responses, hypersensitivity. 3.4 Parasitic infections, protozoa, malaria, Plasmodium spp. Amoebiasis, Entamoeba histolytica. Helminths, Ascariasis, Ascaris lumbricoides. Schistosomiasis, Schistosoma spp. Ectoparasites, scabies, infection by Sarcoptes scabiei mites. 3.5 Parasitological diagnosis, microscopic examination, stool, blood, or tissue samples. Serological tests to detect specific antibodies or antigens. PCR testing to detect parasitic DNA. 3.6 Parasitological therapy, antiprotozoal drugs, artemisinin for malaria, metronidazole for amoebiasis. Antihelminthic drugs, albendazole for worm infections. Prevention, vector control, e.g., mosquitoes for malaria, sanitation and environmental hygiene. 4. Roles of microbiology and parasitology in medicine. Diagnosis, identifying microorganisms and parasites as the causes of diseases. Prevention, vaccination, e.g., BCG for tuberculosis. Sanitation to prevent parasitic infections. Treatment, specific therapies based on the identified pathogen. Research and development, discovery of new vaccines, drugs, and diagnostic methods. Conclusion, The Microbiology and Parasitology course provides essential knowledge about infectious agents that cause diseases and their management. This understanding is crucial for accurate diagnosis, effective prevention, and appropriate treatment of infections in clinical practice.